All right, in this lesson, we're looking at membrane models. So we have a basic understanding of what elements are involved in a cell membrane. Now we've got to understand how that model was constructed. And we've got to remember that in science, knowledge is constructed over a great deal of time with lots of different things proposed. And until new technology comes about so we can understand what things actually look like, there's lots of different ideas based on what a membrane looked like before. So we're in topic 1.3, and our specific dot points today are going to be the analysis of evidence from electron microscopy there and the analysis of falsification of a particular model. Plenty of other things there, but we'll look at them at another stage. Okay, this is our basic membrane model. We're talking about the fluid mosaic. We understand what elements are part of this model. We've got our phospholipids here. Right, and we've got our transmembrane proteins that go all the way through. We've got our glycoproteins or our carbohydrates sitting on top of those proteins, and we've got our cholesterol molecules as well. Now, you are obviously needing to draw a simplistic version of that model. We might make one down the track, but understanding that we can simplify those very complicated 3D models into a simplistic 2D model. Now, back in the day, 1925, GORTER and GRENDEL had their own model. And what they used to do was measure phospholipids in red blood cells. So they would dissolve everything else and just do that. So they actually calculated that the surface area around the outside of that red blood cell, they found twice as much in terms of phospholipids, so twice as many phospholipids as would be required to cover it. So they deduced that therefore there must be a bilayer or a double layer of the phospholipids. And we sort of understand that bilayer exists in our current model as well. All right, the next step up was in 1935, so about 10 years later, and DAVSON and DANIELLI said, right, well, if this is the kind of thing we are seeing, we are going to keep talking about the membrane as being trilaminar or 3 layers. And they proposed a kind of sandwich model, and that's what you see. So as the bread in the top and the bottom there, they're saying that this particular part is a layer of proteins, and then smack bang in the middle where that lighter section is going to be that bilayer of phospholipids. So it's almost like a sandwich with a double layer of meat in between. Unfortunately, these kinds of pictures weren't available to them, and they didn't quite realise what they were looking at. And these kinds of things that make it look tri-layered, we're looking at 2 different cells back to back. So there started to be some issues with this model, particularly. And, if we look at this here with the protein layer on top and the bilayer in the middle, they'd have to say that, right, the proteins that would be in this layer, they would actually have to be the same thickness, so uniform all the way across. And what they started to find was that's not actually the case. This model assumes that it's going to be symmetrical, right? So inside of the cell is going to look exactly the same as the outside of the cell. And we now know, we have the information to say that's not actually the case. This kind of stuff doesn't really explain how some hydrophilic material moves past. So hydrophilic, water-loving. So if you were just a water molecule needing to get into the cell, given that these proteins are hydrophobic, there'd be no way the water would be able to cross that membrane at all, and that's a challenge. Also, scientists could find that the temperatures that the membranes solidified, so they're actually at that kind of shape, didn't match the model itself. So there's a few issues there. So again, this is another depiction of it, and you can see all these purple blobs are the proteins. They started to actually prove with some science that this model is false. So they found that the membrane proteins are not soluble in water, which means they couldn't actually sit on the outside, given that they would be exposed to water solutions. They found that membrane proteins varied in size, which is what we would have assumed. Okay, so they cannot be sitting in that nice, neat sandwich bread part at the top there. They found that proteins were mobile as well. So they were able to tag, say, one particular protein and then follow its little journey. What they found was that it's fluid. It moved around the membrane, didn't just stay put in one spot. And it also found that some proteins spanned the entire membrane. They didn't just sit at the top and the bottom. So there's some real issues. So then what's happened is they've moved across to the SINGER-NICOLSON model. But you can see even these differences here of the years, that's a long time for science to sit with one particular model before going, you know what, here's a better solution. So it does make you think, what kind of models are we working with now that down the track we might actually change our understanding of based on the evidence. So we are currently talking about our fluid mosaic model, which is our Singer-Nicholson model, and it's been sitting there since 1972. And we know that it is protein embedded in the bilayer. So some of these ones span the entire membrane. Some of them are just sitting on the edges there. So these ones would be anchored in different ways. And this is a very simplistic picture, remembering that there would also be some glycoproteins, there would also be cholesterol and things like that. There's some really amazing 3D models out there that you can get your hands on. We'll play with some of these in class, but I want you to remember what we draw as 2D is 3D, and it is covering an entire cell all the way around. So we've got to keep seeing lots of different models of this, or lots of different depictions of it, so we can understand what we are working with. Okay, so remembering that it's called a fluid mosaic. So the fluid part meaning all of this is still able to move around. These are not anchored in one spot. They might actually migrate a little bit. The cholesterol, remembering, slows it down just a little bit because it stops that fluidity. It kind of slows the movement, I guess. And the mosaic part of it is that we have lots of different bits and pieces in here and different elements that make it up. It is not just uniform all the way across. Okay, so remembering we're coming back to these particular points about the DAVSON-DANIELLI model. There are two specific ones once we've got all our other information down about the actual elements in the cell membrane.